Hello again. In this video, I'm going to explain how the prognostic factors in neuroblastoma brilliantly elucidate the hallmarks of cancer. So I'm going to begin by talking about, firstly, nMYC amplifications. So having nMYC amplifications in a neuroblastoma tumor is a very poor prognostic factor because nMYC or MYC feeds into accelerated cell cycle, which leads to rapid proliferation of a neuroblastoma tumor cells, and this is by inhibiting cyclin-dependent kinase inhibitors, which then potentiates the cyclins and cyclin-dependent kinases. So when the cells then progress through G1 synthesis, G2 mitosis rapidly until they become anaplastic, poorly differentiated, and then can metastasize and spread. So as you can see from the first thing, nMYC amplification in neuroblastoma reveals how pro-proliferative factors make a tumor have a poorer prognosis. The second thing is chromosomal abnormalities. So in neuroblastoma, if you have a chromosome 1q deletion or a chromosome 17q gain, this leads to a poorer prognosis. Now, I don't know the exact molecular mechanism behind this, but in general, if you have chromosomal changes, you're more likely to see some unwanted change in a tumor suppressor, where the tumor suppressor stops working, such as p53, or some gain of function in a proto-oncogene, which leads to, say for example, cyclin-dependent kinase. If you have too much of that, or it's defective, then it leads to excess proliferation. The third kind of prognostic factor is telomerase activity. So this is another brilliant, another important hallmark of a cancer, which is telomerase activity. So as cells, rapidly divide, there are going to be changes in the chromosomes. One specific change is in the shortening of telomeres. Now, eventually, telomeres become so short that your cell machinery interprets it as a double-stranded DNA break, and then this leads to quite a natural DNA repair mechanisms. Now, these repair mechanisms usually result in further damage, paradoxically. There's a name for that which I can't quite remember. So if a neuroblastoma has high telomerase activity, which counteracts the shortening of telomeres and prevents this DNA repair, then these tumor cells can continue to rapidly divide and to survive without undergoing intrinsic apoptosis. The next prognostic factor to discuss was differentiation antigens, specifically tyrosine kinase A and B. So if a neuroblastoma lacks these receptors, not antigens, receptors, then they're less likely to receive a stimulus from growth factors that result in differentiation. So the tumor cells will remain undifferentiated, and this is a universal poor prognostic factor. Now, what to explain next is that is to explain the hallmark of cancer, that if it's undifferentiated, it's more likely to be closer to behave like a stem cell, where it's rapidly dividing. But obviously, our body's stem cells are highly regulated, whereas cancers are not. And so they divide and cause damage and spread. The last prognostic factor I wanted to explain was age. So if you have a neuroblastoma diagnosed before the age of 6 months, I believe, you have a much better prognosis than if it was greater than 2 years. And in my previous video, I explained how a pediatric malignancy, which is more close to an embryonic disorder, is more likely to spontaneously go into remission because of the natural history of the tumor. The cells will naturally differentiate anyway as the child grows. But if a tumor has happened begun at an older age is more divorced from an embryonic process and more into an anaplastic process. And this has a poorer prognosis. So what is the hallmark of cancer that's explained here? It is this idea that, again, poorly differentiated tumours, which are more likely to be seen in older age groups, carry a poorer prognosis because morphologically and functionally they're closer to stem cells than they are to well differentiated, functional, well-regulated mature cells. So I hope this video has given you some insights. I'll just summarize what the different factors are. So poor prognostic factors in neuroblastoma include nMYC amplifications, chromosomal changes to 1q and 17q, telomerase activity, tyrosine kinase A and B expression, and lastly, age. I'll hope to see you in the next video. Thank you very much.